Welcome everyone. Today I'd like to discuss 3-mark question and answers in Biology Botany, Model Paper 1. Question number 17 for you. In 4 o'clock plant, a male plant with pale green leaves is crossed with a female plant having dark green leaves. Name the plant type formed in the F1 generation and explain the type of inheritance in this cross. A male plant with pale green leaves is crossed with a female plant having dark green leaves. In this experiment, all the F1 generation plants have dark green colored leaves. This cross is an example for chloroplast inheritance, a kind of cytoplasmic inheritance. In this experiment, the male plant, that is the male gamete, contribute only the nucleus, not the cytoplasm for the F1 generation. The female plant contributes the cytoplasm with chloroplast gene. Then next question for you. What is PBR322 plasmid? You know plasmid, the non-genomic DNA or extracircular DNA present in the cytoplasm of the bacteria. This PBR plasmid, 322 plasmid, is an example for reconstructed plasmid in the lab. This plasmid acts as a cloning vector. Generally, plasmids are also called the vehicular traffic efficient gene exchanging vehicle. So the PBR plasmid acts as a cloning vector in genetic engineering. In this PBR322, P represent the plasmid, BN or represent the scientist names Bolivar and Rodriguez. The scientist developed plasmid, and 322, this number represent the number of plasmids developed from that lab. It is a Bolivar and Rodriguez lab. Next question for you. What is greenhouse effect? Draw the relative contribution of greenhouse gases. You know greenhouse effect, the global warming, the process by which radiant heat from the sun is captured by gases in the atmosphere that increases the temperature of the earth ultimately. The process by which radiant heat from the sun is captured by gases in the atmosphere that increases the temperature of the earth, ultimately called the greenhouse effect or global warming. The relative contribution of greenhouse gases, see the picture. Carbon dioxide contributes 60%, methane 20%, chlorofluorocarbons 14%, other gases 6%. Next question for you. Give an account on cryopreservation. Cryopreservation is a process by which biological materials are subjected to preservation by cooling to very low temperature of -196 degrees Celsius using liquid nitrogen. So it's a process by which the biological materials are preserved by cooling to very low temperature of minus 196 degrees Celsius using liquid nitrogen. The biological materials to be preserved, either cells or tissues or organelles or pollen grains or protoplast or plant parts. In animals, the spermatozoa, the egg, the stem cells, the zygote. So these are biological materials are preserved in this methodology. At extreme low temperature, what will happen? The enzymatic and chemical activity of the biological material will totally stop. It is a kind of germplasm conservation for the future. Right, the differences between habitat and niche. Habitat is a specific physical space occupied by an organism or any species. So the physical space occupied by an organism or by a species is said to be habitat. Same habitat may be shared by many organisms or species, or one habitat with many organisms or a species. Habitat specificity, not specifically specificity, is exhibited by organisms. Niche refers to an organism's place in the biotic environment and its functional role. Each and every organism has a role to perform in the ecosystem. So organisms place in the biotic environment and its functional role, either a producer or a consumer or a decomposer, is important. A single niche is occupied by a single species. And finally, organisms may change their niche with time and season. Thank you, students. Thanks a lot.